Good afternoon. In today's lecture of computational botany, we are discussing about sequences used in bioinformatics. In previous lecture, we are discussed two types, that is genomic DNA, cDNA, that is complementary DNA. In this, remaining three sequences, that is organellar DNA, expressed sequence tag, and genomic sequence tag. Here, two types of organellar DNA, that means chloroplast DNA and mitochondrial DNA, that is cpDNA and mtDNA. So see one by one, that is chloroplast DNA. In chloroplast DNA contains 20 to 200 copies of circular double-stranded DNA molecules, which may be also linear, which is found in maize. That means the chloroplast DNA is found in this maize plant. Here the short single copy and long single copy of the chloroplast DNA molecule. It contains the 10 to 20 kb long and carrying ribosomal genes in this chloroplast DNA. And in shorter single copy contains 18 to 20 kb long. You can see here three parts, that means the short single copy and long single copy, and the black section containing the chloroplast DNA. There are some opening regarding the frame, that means ORF, which coding the sequences beginning with the met codon and stop codon at the end of chloroplast DNA. Chloroplast DNA is relatively large, that is ranging from 120 to 10 kb, which is comparable to the size of large bacteriophage, which is 110 kb. So the total chloroplast DNA in cell may contains 40% cellular DNA. Out of 100, which is containing 14% present in this chloroplast DNA. In most chloroplast genome appears to possess the same set of genes. That means each molecule of chloroplast DNA is encoded with 110 to 120 genes and codes for about the 120 proteins. In maize, the gene of chloroplast contains both small and large RNA genes and sequence for the tRNA. It is also synthesized mRNA for the some proteins involved in the synthesis, like enzyme riboswitch, which is involved in the photosynthesis in plant. Here the second one is mitochondrial DNA. Like as a chloroplast, it is contain 5 to 100 copies DNA molecules, which is usually circular, but may at the essential it is linear. So you can see in this diagram the remarkable variations exist in the size of mtDNA, that is mitochondrial DNA, ranging from 10 to 2500 kb. Here in mitochondrial DNA contributes only 1% cellular DNA, except in yeast it contains 80%. Here this diagram containing the yeast of, that means the Saccharomyces cerevisiae. In this mitochondrial DNA contains different regions which contains ATPs or RNAs. In this, the black circle containing the mRNA or rRNA synthesis. In mitochondrial genome displays the greater variability in gene content. That means the different variability occurs in this mitochondrial DNA. The cloning and sequencing of the entire mitochondrial DNA have been made in several organisms including humans. So it contains gene of RNA, rRNA and tRNA ribosomes associated with proteins, the mitochondrial proteins and enzymes. In this, the substantial fraction of the mitochondrial DNA in each represent unidentified readings frames containing introns of split genes and appears to code for protein required for the mRNA splicing. Splicing means the in previous lecture we can see the about splicing means the exon and introns. In that intron region can be eliminated or only exon regions are take part in the synthesis of complementary DNA. That means exon contains coding region. In this, in higher plants, small circular, sometimes linear also DNA molecule present in addition to the main circular mitochondrial DNA. In this, the main circle contains the repetitive sequences, and due to extensive recombination between these repetitive smaller molecules originate. There are, when in the main circle, there are excess sequences occurs. So there are repetitive sequences which forms the smaller molecules, and it originates from this the main circle which forms the smaller circles which containing mitochondrial DNA. In this, the main interesting feature in mitochondrial DNA is unidirectional and highly asymmetric replication. There is no any symmetry in the replication when it goes to a replication process. In human, it contains 1.2% of mitochondrial DNA. Of the DNA in vertebrate genome encodes the protein. Here, the third one is expressed sequence tags. In this, there are short nucleotide bases, that means 200 to 500 nucleotides. The DNA sequences that can be used to identify a gene that is being expressed in the cell at a particular time. Here, the procedure of how to express sequence tag. In isolate the messenger RNA, that means mRNA from particular tissue. For example, liver, that it with reverse transcription. In when you know the process of reverse transcription. In previous lecture we have discussed about the reverse transcription. That means the process of producing complementary DNA from the mRNA. Here the reverse transcription in DNA polymerase that uses RNA as the template. Thus it able to make genetic information from the reverse RNA. For example, here from complementary DNA to expressed tag. In sequencing, only the beginning portion of the complementary DNA produces what is called as 5' expressed tag. Expressed tag, that is EST. Here you can see this diagram, that is expressless sequence tags are generated by sequencing of complementary DNA. That means the isolation of mRNA from the tissue, and here takes place the reverse transcription process by the using reverse transcription enzymes. Here the mRNA are reverse transcribed to produce complementary DNA using these DT primers. Here take place one strand of RNA which containing the nucleotide pieces which form the another complementary DNA. It contains only codon regions, that means the exons. Here the ribonuclease degraded of RNA and synthesis of second strand of DNA, that is double stranded DNA which contains only exon regions. So here use of expressless sequence tag to sequencing of complementary DNA. Here last one is gene sequence tag. In this there are short, that is 200 to 500 base pair containing DNA sequences occurs, but once it can genome and whole location and base sequence are known. Here the sequences which are known to the base pair which containing base pairs. Here that STS means the sequence tag site are detected by the polymerase chain reaction, which are useful to localizing and orienting the mapping and sequencing data. In this, here you can use the gene sequences for different purpose which containing, that means it construct the genetic and physical map for sequencing and reporting from many different laboratories. Second point is the sequence tag site, that means the gene sequence tag it contains genetic polymorphism, that means simple sequence length and polymorphism, and single nucleotide polymorphism. They become soluble genetic markers. Here the third important use of this gene sequencing, that is it used to screening by polymer chain reaction to detect micro deletions in the azoospermia, that genes which is infertile in man. This is important use of this gene sequence tags in bioinformatics. Here the end of this sequencing which are used in bioinformatics. In next lecture we are discussing about the statistical analysis and evaluation of blast result. Okay, thank you.